When you open the Orbit Targeting Oligo Design app for the E. coli MG1655 genome, you will be greeted by our favorite GalK locus displayed here in the Genome browser window. The fastest way to interact with this app is to go to this drop-down and select your gene of interest. For example, we'll go to HIS-A, which is a gene that we studied in the Orbit Nucleic Acids research paper. So when we click on this, the entire app jumps to that new genomic position, and we can continue to interact with this genome browser window by, for example, clicking on genes in the flanking regions to the left or right. We can also use these arrows that are set to jump by a certain step size to move, for example, 1 kb left or right. And we can also go here and specifically type in a genomic position of interest and jump there. And so you can see that as I'm jumping around the genome and selecting genes, the app is auto-magically generating this targeting oligo sequence here from 5' to 3', which is how you will order it directly from your DNA synthesis provider. And I want you to notice that in this case we have the type of targeting oligo, or TO, set to deletion, and we can easily verify that for this forward strand gene in this replicore because we can see the start codon as the final bases in this homology arm, and the stop codon is the final bases, or the first bases in this homology arm, so this oligo deletes everything in between. However, do not be alarmed if it is not this obvious and blue and orange do not match. The simple explanation is that when we go to a different gene, if we go back to GalK, orange is now on this side, blue is on this side. That is because the targeting oligo is coming from the minus strand, but it is still displayed as a 5' to 3' sequence. So, when we're designing our targeting oligo, there's a few parameters that we can set here. The first is homology arm length. The default is 82 base pairs of homology, plus this middle 38 base pairs gives a 120 nucleotide oligo. We commonly order those from Sigma Millipore. We've also used 90 nucleotide oligos that are 52 base pairs of homology that we commonly order from IDT. This is not an endorsement. Feel free to use whatever DNA synthesis provider you want, but when you're ready to order, all you do is click copy targeting oligo, and this sequence is on your clipboard. So, the next parameter here is @B direction. We'll talk about this more later when we, in another video, when we discuss this tab, integrating plasmid output. But for now, you can see that the @B direction refers to the genomic direction of @B, and in this case, the forward sequence is displayed here, and when we do minus, it just puts it in the opposite direction. We have some preset types of targeting oligos that might be useful for you, deletion being one of them. Which just again deletes everything between the start and stop codon. Note that you can carefully adjust these. For example, if you wanted to delete outside of the start and stop codon, you can move things back and forth very carefully or input specific genomic coordinates there. We also have some different options for you, including C-terminal fusions, which need to be paired with the correct orbit integrating plasmid and need to be carefully designed, but just to show you what that looks like, when we select that gene, the integration is actually coming right there before the stop codon TAA. Final option is insertion, which is not particularly useful, but might be an interesting starting point if you want to design something in the middle of a gene. So that is the basics of how to use the Targeting Oligo Design app to get your targeting oligo. In the next video, we will discuss how the design of this targeting oligo affects the final orbit integration locus structure.